The overall goal of this procedure is to characterize biofilm interactions with the surrounding environment using integrated methods. To do this, a double inlet microfluidic flow cell system is constructed in which glucose-containing and glucose-free media are pumped across a flow cell, generating a glucose gradient. Bacteria are added to the system, and biofilms are allowed to develop over the course of several days. The biofilms are then imaged by 3D confocal microscopy. To characterize solute transport within biofilms, fluorescent tracers are injected into the flow system and time-lapse imaging is performed. To characterize the flow field around biofilm colonies, fluorescent microbeads are injected and time-lapse imaging is performed. Solute transport and particle tracking analysis of the resulting data demonstrate the utility of this method in assessing biofilm processes under various chemical environments within a single device and in one set of experiments.
The main advantage of these methods is that they allow for the simultaneous assessment of multiple aspects of biofilm environment interactions.
The methods reported here can help answer key questions about biofilm formation in natural environments or in biofilm-related infections.
Working in a laminar flow hood, transfer a colony of P. aeruginosa, PaO1 GFP, and a colony of E. coli DH5-alpha from an LB plate to separate tubes containing 3 milliliters of LB broth. Incubate the cultures overnight at 37 degrees Celsius while shaking at 225 RPM. This flow system is based around a double inlet microfluidic cell that facilitates observation of biofilm growth under a well